This sequence shows the anatomy and movement of a very important organism in research biology, the nematode worm C. elegans. This sequence shows the beating heart, the brood pouch containing eggs, and the characteristic saltatory swimming motion of the water flea, Daphnia pullix.
This larva is in the process of molting, shedding its first instar larval cuticle, spiracles, and mouthparts as it becomes a second instar larva. The larva pierces the old cuticle with its new set of mouth hooks, opening up the anterior end. Under higher magnification, the new and old mouthparts can be easily seen. With its posterior end free as well, the larva moves back and forth within the old cuticle. Higher magnification shows the new posterior spiracle. Eventually, the larva backs out of its old cuticle, leaving the old cuticle and mouthparts behind. Following head eversion, the pupal stages begin. The pupal cuticle separates from the puparium, making it possible to dissect a pupa from its pupal case. In the first pupal stage, as seen from the side, a translucent patch is visible in the developing eye. Legs and wings extend ventrally. In this slightly older pupa, the Malpighian tubules seen in the abdomen have turned from white to green. The pupal cuticle looks like a clear bag enclosing the pupa. As metamorphosis continues, as seen in this pupa being removed from its puparium, eye pigmentation begins to develop. Eyes are first light yellow. They darken to amber. Eyes become bright red. Bands of thoracic muscles are visible. Malpighian tubules are bright green anteriorly and yellow posteriorly. Later, the tips of the wings become light gray. In ventral view, sex combs are visible on the male's forelegs. Wings then darken to black. The head, thoracic, and abdominal bristles are now black. Claws on the tips of the legs are visible. At the end of metamorphosis, the adult fly, fully formed within its puparium, is ready to emerge. The legs twitch in preparation for this event. Eclosion, the emergence of the adult fly from the puparium, begins with muscle contractions. The operculum, a door in the puparium through which the fly will exit, opens. It is forced open by a sac on the front of the fly's head called the talinum, which expands with blood to break the seam of the operculum. The talinum continues to expand and contract. Abdominal contractions and legs are used to exit the puparium.
The complete metamorphosis of butterflies and moths is regulated by hormones. As a larva grows, it molts several times, a process mediated by the hormone ecdysone. In the presence of juvenile hormone, each molt results in another, usually larger larva. Juvenile hormone is released from the brain in declining amounts as the larva matures into later instars. When the level of juvenile hormone drops, the final mold into a pupa occurs. Once the larva is secured in place, it sheds its skin, revealing what will become the hardened shell of its pupa case or chrysalis. The release of juvenile hormone ceases during pupation and allows for metamorphosis into the adult or imago stage. In this sequence, a monarch butterfly demonstrates the emergence of the imago from the chrysalis. When ready, the adult breaks through the chrysalis with wet wings. Over the course of several minutes, the butterfly uses the help of gravity to pump fluid from the body into the wings in order to expand them to full size. This real-time sequence shows the flatworm mesostoma capturing, consuming, and digesting its prey, the crustacean Daphnia. In these real-time sequences, two species of rotifers, Floscularia and Colotheca, are shown feeding by creating currents that sweep prey organisms into their maw. In this real-time sequence, multicellular animals, rotifers, are feeding, sucking prey organisms into the gullet. The high magnification sequence shows ingested cells being crushed by plates at the entrance to the stomach. These real-time sequences show the bryozoan Plumatella feeding using water currents created by the cilia lining its tentacles.